Hi students, welcome to Turito live classes. Myself A. Swagat. Today we are going to discuss the last topic of the chapter, cell, the fundamental unit of life. Here we are going to look at the topic cellular organelles, which we have left out in our discussion. After doing that, we will be going through the prokaryotic and eukaryotic cells. Now, before going to that, we will be having an understanding of the topics what we are going to learn in here. Looking at here, we will be exploring some of the topics such as vacuoles, which were left out, and a detailed understanding of microbodies. So, understanding the functioning of the organelles. So, we will try to look at their structures, possible structures, and we will be understanding their functions, what exactly they are doing in the cell. Once done, we will be looking at the complexities of different cells using structures. So, we have been doing this particular thing since quite a long time. That means, we are trying to understand cells. So, as you know, we have different types of cells. Okay, that is the bacteria, fungi, plant cell, animal cell. Which one is highly complex and which one is so simple? So, we are trying to understand that in this particular objective. Once done, we will enlist all the complexities and we will compare the complexities between prokaryotic and eukaryotic cells. So, this is what I have planned for you for this particular session, children. So, let us move forward and learn ahead. Before going to that, we will recollect what we have already learnt. So, in the previous session, we were having a detailed understanding of Golgi complex, the structures, lysosomes, centrosomes and plastids. So, we learned these things using the images what we have, and after that we try to understand their structure and observe their functioning, what do they do inside the cell. And then we used a flow chart where we try to look at different types of plastids present in the plant cell and their respective functions, thereby understanding their significance within the plant cells. So, that is what we understood. So, commonly we know them as plastids, and we saw that there were different types, amyloplast, alloplast, leucoplast and all those types of plastids. Now, here today's session we will actually start with one of the important organelle of plant cell, that is a vacuole. So, when you look at it, just like many organelles what we have learned so far, leaving few exceptions, this particular vacuole is one of the organelle present in the plant cell. It is having a very big in size. Okay. Following that, we also find that in fungal cells, and we also find it in animal cells on a temporary basis. Now, these are membrane bound organelles. So, just like many other organelles, even this particular vacuole is covered by a membrane. So, they are temporarily found in animals, as we all know, like examples like animal like organisms like amoebae, paramecium, euglena, stentor, there are so many. They may find them in for a limited period of time, that is a reason for them. And this one, the vacuole occupies 90% of the cell volume. Can you tell me in this particular statement which cell is referred here? Okay. So, when I see here you guys are dropping in the correct answer here, when it is said 90% of the cell volume is occupied, it is referring to the plant cell. Okay. So, that is where you see 90% of the cell volume is occupied by this vacuole in the plant cell. Okay. Now, we need it because it provides turgidity to the cell and rigidity to the cell. Turgidity refers to, when it comes to plant cell, especially mentioned there, okay, it refers to, you will understand more by looking at the structure, but before looking at the structure, let me tell you something. As you all know that it occupies 90% of the cell volume in the case of plant cell. So, it contains the cell sap, most of the cell sap is present inside the vacuole in the case of plant cell. Fine. What does that cell sap contains? It contains obviously whole lot of amount of water. Now, when it contains whole lot of water in that, now you must understand it is such a component present in every cell, every living organism. When it comes to vacuole, it also contains water, that is what I said. Along with water, it has many solutes within itself. Because of this excess water, okay, it increases its size anyway, and that vacuole which is having a lot of water puts a whole lot of pressure on the cell wall. On the cell wall of the plant cell. Okay. Now, when the cell wall is under pressure, you must understand previously we were trying to understand the effect of different types of solutions of an animal cell and a plant cell. So, when it comes to plant cell, you saw irrespective of the type of solution, the shape of the plant cell was intact. Do you remember? If you remember, the reason was cell wall. In this case, let me draw a structure. Consider this to be the plant cell, okay, and in that if you see, most of the region is actually occupied by the vacuole. Now, this white in color what I have drawn, consider that to be a vacuole. If that is the case, you must think that it also exerts some kind of pressure around itself because it contains water, and when it comes to this yellow border, consider that to be the cell wall. Equally, cell wall also it exerts pressure on this organelle, thereby keeping the shape intact. The shape of the cell wall would be intact, and ultimately the shape of the cell is intact. So, there will be no change you finding there. So, that is what we find this one is having the particular rigidity, that is because of the pressure, rigidity, that is because of the wall pressure ultimately. So, ultimately overall the cell shape would be intact and the organelle would look like this. Now, this is the organelle what you are looking at, and this is how the cell vacuole would look like, that is plant cell. Like it is mentioned earlier, it is the having a membrane surrounding it, is not it? That membrane has a specific name, that is tonoplast. Tonoplast is the membrane which actually bounds the cell sap of a vacuole. So, here whatever it is containing inside it is known as cell sap. Any questions by now? Okay. Let me tell you something more about it. Now, what is the use of this big vacuole in a plant cell? There are so many uses. Based on your previous understandings, can you get me some of the uses of vacuoles in a cell? Okay. Okay, great. So, let us see. Some of the uses are like this. One of the use is it helps in digesting food. Okay. So, when we say digesting food, obviously we are thinking about an animal cell, and of course, we do find the same process in some of the plant like cells. Okay. Now, digesting food is one such thing. Apart from that, you see storing wastes. So, whatever activities are happening in the cell that generates some type of wastes or the byproducts. Byproducts are those products, they are formed because of the reactions occurring in the cell, and those products are generated, but then they are of not use to the cell. Those products are called byproducts, they are just stored inside the vacuole. Okay. Now, that is one such reason. Now, if you remember amoeba, it was also using the food vacuole, digesting all the food inside that vacuole, and ultimately that was combining together to form another vacuole, a bigger vacuole in size called contractile vacuole, which actually uses as waste discarding vacuole. So, contractile vacuole in the case of amoeba was a waste discarding vacuole. Okay, that was good, great, fine. But here also you will find in the case of plant cell, you will find the same thing. It will be consuming so many activities happening in that. So, it it generates waste and that will be stored inside it. Okay. So, that is also given out. Apart from this, let me tell you something else. There are some plants, children, you must understand. When you look at those plants, okay, you may look at it as a common plant, that is fine, but then they do have so many vacuoles within each and every cell of their plants, okay, of the body. There are few herbivores, you might have observed, they do not bother some plants to consume. Have you ever thought of it? Why? Okay, I see some response. So, the reason behind that is, some of the herbivores, if they try to consume those plants, those grasses, some of the grasses, they cannot consume it because there will be some kind of spine like things or the hairy like projections will be there on the surface of the grass. So, that will not help those herbivores to consume them, or there will be some chemicals released by the surface of the leaves. So, that when a herbivore is trying to consume it, it will have some kind of itching sensation, okay, the whole of the mouth and the tongue gets irritated, itching sensation. So, that is one of the reason why they do not consume. There are other category of the herbivores, they do not consume some type of plants. The reason is this is the reason. Now, how this can be reason, I will explain you. What happens there is, in some of the cells of the leaves, okay, of some of the plants, what do they contain is sometimes, okay, it is not visible. Some type of chemicals, okay, in their vacuoles, okay, some type of chemicals in their vacuoles. Now, what happens with that is, when a herbivore, suppose a deer or a impala or a black buck comes and try to consume a particular type of plant, okay. Now, what happens, those chemicals which are directly coming out from the plant, okay, that is especially from the leaves. You might have seen when you tear a leaf, you will see some watery liquid comes out, or sometimes a white milk like liquid comes out, is not it? Similarly, when these herbivores are trying to consume that particular plant, they too release some kind of liquid coming out of their thing. At the same time, you will see liquid which was stored inside this particular vacuole, potentially consuming, potentially having some type of chemicals which actually irritate the tongue, having some kind of too much of irritating sensation within them. So, what do you and me, for example, if we are having that kind of sensation, we will end up using our hands and try to pacify that thing. But how do these animals do that? So, what do they do is, next time they will not bother that particular plant, and that is what this vacuole will be containing some chemicals, and that will be released and going into the mouth of those herbivores, and that is how they will not try to bother that some of the plants. It is also known that some of the vacuoles of some herbs contain cyanides, some type of cyanides, not the poison one, but they do have. So, this is one of the use what we can find in there, used as one of the beautiful defense mechanism in the case of plants. Hope you got the point. Okay, then let us move forward. Okay. If you have any questions, please drop in and try to get back to you with the answers in a short while. Okay, let us move forward then. Here. So, I was also mentioning about contractile vacuole, food vacuole. So, if you look here, this is one of the one of the organism which we are quite aware of it. It is a kind of one which we actually cannot forget at all. So, here if you observe, this structure, what it is forming, a pseudopod. Okay. Two of them, obviously that is pseudopodia. Pseudo means false, podia means feet. Okay. Now, here when it is trying to consume this one, it engulfs it. So, ultimately what happens? Let us say this one be the first step and you see this to be second step. Okay. That is quite simple. Now, what is this? This what you are looking here is nothing new to you, which is actually the food vacuole. When that is the food vacuole. So, you would see certainly it will be remaining in the cell of this particular organism as long as it has the nutrient component within itself. So, it will take some time for this organism to digest this food vacuoles which are containing foods, is not it? See, all of them, they are containing some sort of food within themselves. So, that has to be digested. Once that food is getting digested, after the digestion process, you would see all that waste which is generated within them. So, surely here, here and here, you would see once the food is getting digested, in those 4 vacuoles waste will be generated. It is obvious, is not it? Now, when that waste is generated in this organism, you will see that this organism would be putting all those waste, trying to collect at one place and dump it in another vacuole, that is contractile vacuole. So, that is where it will be dumping all the waste. Now, when the waste is dumped there in this particular vacuole, that is contractile vacuole, amoeba uses that vacuole to discard it, throw out that waste from the body of this organism. So, that is how it happens. We do find vacuoles in animal cells also, but they are temporary in nature. They are available in the cell for a limited period of time. So, reason you just understood. Okay, that is great. Any more questions you have? Okay. Now, let us have a look, like I mentioned, micro bodies. Now, these micro bodies are also organelles present inside the cell. Probably you might be wondering, we have never heard of it. Surely you might never have heard of it, but I just wanted to put it forward to you, like these are the things which are also available in the cells. They are nothing but, they are the some of the projected, dilated or extra lengthy parts which were detached from some of the organelles they are formed. Here about micro bodies, when we are trying to understand, you must realize that by looking at it, there are some of the observations recorded here. Let us read them and you will understand now where do we find these micro bodies, how they are originated in the cell. So, that is what we are trying to understand now. So, there are observations that micro bodies originate in 3 different ways. That means, how do they form inside the cell? Firstly, there are dilations in the tubular ER. ER stands for endoplasmic reticulum. Now, what is dilation? Dilation is something, for example, if there is a vein like this, okay, this is a normal vein what you can see. If some says, because of their observation, they said that the vein is getting dilated, now that is our understanding. I will show you. So, you observe here that when the people say that vein is getting dilated, can observe this region, it shows that that is a dilated region. Okay. So, that can be like this also, that is a possibility, that is also a type of dilation. Okay. So, it is said that it is also a dilations. Okay. I have gone wrong here. Okay, now it is correct. Dilations. Okay. Now, that is what is dilation means. Here it is said about endoplasmic reticulum getting dilated. So, from that endoplasmic reticulum, which is tubular in shape, having, if that gets dilated, you would see that dilation would be pinching off from the ER, leading to formation of microbodies. That is one way of forming that particular structure. So, that is one way of forming that particular structure. Secondly, as lateral buds from opposite sides of ER cisternae. Now, ER cisternae means, if you remember ER, you know, it was having a specific structure, okay, it was having two types of structures. At the end you will see from either ends, I would like to draw it, okay, here. Okay. So, if you observe here, okay. So, if you observe carefully, the ER would look like this. Okay. So, it was having different structures, okay. There were few dots available, something like this. Now, these projections are common, quite common in the ER. When you see those kind of projections which are quite common in the ER, sometimes, you know, at this juncture you see some projections are formed, sometimes here. These extra projections, you know, that will be given off, or these buds would be pinched off. That pinching off would eventually lead to the formation of microbodies. That is the second point. Thirdly, you see there are already available microbodies, that means microbodies have already formed. Okay. When some of the microbodies are quite long and tube like structure, you will end up seeing them dividing into two parts. That would lead to the formation of further microbodies. So, these are some of the ways how it is formed. So, hope you understood how it is formed in the body of a cell. Now, you see these microbodies are kind of organelles present in plant cells, animal cells, protozoa. Obviously, wherever you see some of the organelles like ER, endoplasmic reticulum, you will see the formation of microbodies. That is a usual scenario. Here you see they have specified these are the cells where you will find the presence of the microbodies. Now, it is not all about microbodies, we still have a whole lot of things. That is, if you observe, microbodies are classified. There are different types of microbodies, starting with this. If you observe, microbodies are of 4 types, namely peroxisomes, glyoxysomes, glycosomes, hydrogenosomes. So, hope you are understanding the concept is quite clear here. Microbodies are leading from the already available organelles, and they can also form from the already available microbodies. Okay. They are of 4 types: peroxisomes, glyoxysomes and glycosomes, and then hydrogenosomes. Now, we will be looking into them one by one, and we will understand what do they do in the cell. If you look here, first one is quite simple, that is peroxisomes. What does it do in the cell? Peroxisomes are mainly present in the cytoplasm, okay, and they have an important function. If you remember something, you can link it with something. detoxification of toxic substances. Now, children, tell me, from where did we get this detoxification of substances as a function performed by peroxisomes? Any guesses by you? How this guy is doing that? Someone else was doing that, right? And some other organelle in the cell was doing that function, but this guy is doing it now. Okay, as you have rightly spotted, as we have seen, these are the some of the microbodies were actually originated from ER, particularly SER. So, when you look like that, then you will understand clearly that it is actually the smooth endoplasmic reticulum which was giving rise to some of these microbodies, that SER was also having this function, detoxification of substances. That is why it is having it. Following that, it also performs a process of photorespiration. So, it will also perform the process of photorespiration. So, that is more important. So, hope you got my point about peroxisomes. Next is glyoxysomes. This is nothing difficult for you children. Okay. Specialized peroxisomes in plants is known as glyoxysomes. So, it is the same thing, but in plants it is known as glyoxysomes. Glyoxysomes are present in plants and they perform the function performed by the peroxisomes. That is the same thing. When it comes to glycosomes, glycosomes are also peroxisomes. They are present in protists. Protista is a group. Protista is a very big group having all unicellular organisms and exhibiting animal like characters. That is what these kinds of organisms you find in Protista, and of course, you may find some of them having plant like features also. That means exhibiting plant like or an autotrophic features also. That is what the group is all about. Now, finally, you see hydrogenosomes, and this one is something different and typical. When it comes to hydrogenosomes, this is an exception in the microbodies list. Why? Because found in anaerobic eukaryotic organisms. Anaerobic means, what can you say about it? Anaerobic eukaryotic cells. Great. So, you see, anaerobic eukaryotic cells means those organisms which actually they do not use this thing for their survival. They do not need that for their survival. That is what is anaerobic, guys. Fine. Presumably, they think that this might have evolved from mitochondria to produce molecular hydrogen and ATP in anaerobic conditions. Now, that is interesting. This particular hydrogenosome, that means this particular microbody, it is thought that it might have generated from mitochondria. That is a contrasting thing. I know I am saying that anaerobic organisms and again saying that mitochondria. That might be a bit contrasting, but you must understand why they are saying, because this microbody has the ability to generate hydrogen in the form of H2. So, H2 is not an atom, H2 is a molecule. That is one reason. Second reason is it keeps the ability to form ATP. Now, where do you get to see this ATP formation? As you have rightly dropped in, we get to see the ATP formation in mitochondria. That is where we see that. Okay. So, that is the reason why it is understood that this hydrogenosomes are one kind of microbodies presumably evolved from mitochondria, that means originated from mitochondria, as they produce molecular hydrogen and ATP, that too in anaerobic conditions. So, that is some important reason you have to understand. So, this is all about microbodies. Hope you understood the concept. Any questions by now? Anything you want to mention? Okay. So, looks good. Let us move forward. The topic what we are to understand is this: cell in relation to complexity of body organization. Now, this is actually the final understanding what we will be having in a while. Okay. It is all about the cell, how complex it is. Okay. There are cells which are present in different organisms. You know, Monera being the group having all the bacteria within itself. So, you can understand that that is actually the prokaryotic type of cell. That is good. When you think of cells in our body, our plants or fungal cells, they are basically from the eukaryotic type of cells. So, by now you might have clearly understood what we are going to understand now. That means, our upcoming discussion will be actually based on the comparison of two types of cells, namely prokaryotic cell, okay, followed by eukaryotic cell. This will be our topic of discussion shortly. So, when it comes to prokaryotic cell, you see the basic features what you know since class 8th. Some of you might have read in class 7th also probably. So, if you observe carefully, we would end up understanding prokaryotic cells have pseudo nucleus known as nucleoid. Okay. So, it has a pseudo nucleus commonly known as nucleoid. Pseudo, okay, it is written wrongly here, okay, fine. So, that is false nucleus. It is performing the function of nucleus, but it is not actually nucleus. This kind of thing we would find in prokaryotic cell. Okay. Hope you got it. This is what we get to see in the prokaryotic cell. Okay, that is good. Now, you see that you will not find much of the organelles in them. Okay. And even if you find them, you will not see they are you will see that they are not surrounded by any sort of membranes. The best example is bacteria. When I say bacteria, there are so many types of bacteria. There are different shaped bacteria you have learned in the lower grades, starting with cocci, bacillus, sorry, what was that? So, cocci, bacillus, vibrio, spiral, there are so many types we have seen. These are some of the groups what you can see. So, this is in short about prokaryotic cells. Coming to this, what we have in our body, what plants have, what kind of cells does the fungal have, fungi, the mushroom is what we consume. Okay. So, we get to see the presence of true nucleus. So, in our system, this is quite clear to us that when we have a very true nucleus, it is not a false one, not the pseudonucleus, not the nucleoid in case of bacteria. In our body, we have a true nucleus in each and every cell. If that is the case, you will see nucleus is enclosed by a membrane. So does the different organelles present in the eukaryotic cell. So, even the organelles present in the eukaryotic cell, they will be having membrane surrounding them, with some exceptions like ribosomes, they do not have any membrane covering them. Now, here it is mentioned about plant cells, animal cells, fine. I would like to add fungal cells also. They are also coming to the very same group, that is eukaryotic cell type. This is in brief to make you understand that this is what we have been going through in the lower classes. Now, we would like to have a detailed understanding by looking at some of the points like this. Prokaryotic cells having a size less than 5 micrometers. So, for your understanding, 1 micron is equals to 10 to the power minus 6 meters, and they are always, you will see them having only one cell as an organism. That is why they are always unicellular, following which no nucleus instead has a naked loop called nuclei. That means, they do not have a true nucleus. We just discussed they have a pseudo nucleus. It is not like a loop. Loop means this is a kind of a loop. Okay. When it is said it is a naked loop, okay, it is not like a true one, it is just circular one and available like that, it is something like this, and it is not having any covering over the nucleus. If there is a disturbance occurred through the whole the body of the cell, you would see the contents here would be easily suspended in the cytoplasm of the organism. So, that is why you call it as a nucleoid, following which you see that having organelles which are present there without any membrane. They do not have any membrane covering themselves. That is the case only with the prokaryotic cells. You see, ribosomes are small. So, size of the ribosome in the case of prokaryotic cells is quite small, that is 70S. S stands for Swedberg unit, a measure of the size of the organelles. Particularly about ribosomes, the other day we discussed very clearly how this Swedberg is used to understand, which actually gives us the indirect measurement of the size of the particles which are as small as ribosomes. No mitochondria present here. So, you would see them having no mitochondria, and cell division is by binary fission. So, binary fission is one of the method where an organism of one cell type would end up dividing into two cells like this. So, one cell would give rise to two cells. So, that is quite simple. And then reproduction mainly asexual, that is binary fission is one of the method of asexual reproduction. Okay. And you see here there are some non-meiotic forms of gene exchange such as conjugation. Conjugation is like when there are two different organisms in the unicellular type, they try to combine together, they try to mix up the nucleus. That process is referred here as conjugation. So, that is one such possibility where these prokaryotic cells would do in order to change their genetic composition. If there is a change occurring like that, it is marginally understood more or less like sexual reproduction, that is meiotic forms. Okay, that is what I want to tell you. So, many metabolic pathways, fermentation, nitrogen fixation, photosynthesis are occurring here. In this particular one, you would see them happening that, like I mentioned. These are all the types of bacteria what you would see, and they are all of prokaryotic types. Okay, they are prokaryotes. So, bacilli stands for rod shaped, chain of the rod shape you see, and you would see spherical shapes of spheres, coccus, cocci, diplococci, chain of cocci, cluster of cocci, and we do call them as bacteria. Mostly they do not live as free, most of them they live as colonies. Okay, spirals, spirillum, spirochete. So, these are some of the common things what we can understand. Okay. So, this classification of shapes is also not new to you, because we have already learnt in class 8th. Okay, that is where we have seen. Now, this is a typical bacteria cell, bacterial cell, showing us all the organelles and different parts of it. Let us have a look at them. Okay. Starting with the outer covering having hair like projections known as pilus, and that covering is known as capsule. So, that is the covering what you see known as capsule. Okay. And then you see that it is having another layer called cell wall. If, let me tell you, hope you are following my understanding, my discussion. If you want to enter this particular bacterial cell, for the first time you have to go through the capsule, secondly you will enter the cell wall, and the third way you would enter the plasma membrane. After crossing the plasma membrane, you would see that you will be freely suspended in the cytoplasm, and that shows us that we have so many types of organelles are present. I mean, some of the organelles are present inside it, and then you would see the presence of the nucleus. So, this is what is the pseudo nucleus it was referring to. This is how the nucleus in the case of prokaryotic cell, right. So, you see there are ribosomes available, yellow in color. There is another organelle called plastid, there are small mesosomes, pilus, and this tail like structure over here known as flagella. This tail like structure over here helps this whole organism try to navigate inside the aquatic medium. So, mostly the medium would be water. Okay. If it has to navigate there, it needs flagella, whip like movement happens there, and that is how this whole body is propelled through the water medium, or in other words aquatic medium. So, hope you understood. Next topic of our understanding is eukaryotic cell. They are quite large cells, comparatively very large cells. In fact, they are actually larger than 10 to the power minus 6, 10 into 10 to the power minus 6 micrometers. That means 10 micron, more than, in the least size is actually more than the 10 micrometers, and often multicellular in organizations. So, most of them are multicellular in organization, okay, and you would see there are also some of them which are of unicellular type. Okay, there are also unicellular type. Always has nucleus with linear DNA containing chromosomes and histones. Histones are proteins, over which you will see that the DNA chain is binded by. Okay. So, they have a nucleus with a linear DNA. In the previous case, that is for the prokaryotic cells, there the DNA was kind of circular, it was a loop. Here it is a linear DNA like a single chain. Membrane bound organelles. Most of the membranes in the case of eukaryotic cell, they are confined by a membrane, and that membrane would have the same chemical, more or less the same chemical composition like that of the plasma membrane. Okay. That is a part, and then ribosomes are large in the size. So, in the case of eukaryotic cells, ribosome size is quite large, which as in the case of prokaryotic cells, in prokaryotic cells you saw the ribosome size was 70S type. Okay. Now, if you remember, when we are trying to understand the 80S and 70S type of ribosomes, can you just get me that what are its bifurcations, if you remember? And that is quite simple. If you do not remember, let me tell you that 80S comes up with the larger unit as 60S and the 40S, okay. And here the case was like 50S and 30S. So, the 60S stands for larger subunit and 40S stands for smaller subunit. Same is standing for prokaryotic cells. This is what we learned when we were trying to understand the organelle ribosomes. Okay. Next is, here you will see the presence of mitochondria in the case of eukaryotic cells. So, hope you understood this. And there we saw the division was binary fission, part of them were also exhibiting meiosis, but here you would see cell division is by both mitosis and meiosis. Thereby I am saying they are relevant to asexual, sexual. So, This is a type of reproduction occurs in eukaryotic cells. Common form of respiration involving organic molecules like sugar. So, when sugar is needed is to generate energy. If it has to generate energy, it needs oxygen. Okay. So, oxygen and sugar gets converted, combined together to generate energy, and that is why this one is available here, that is mitochondria, which is known as powerhouse of the cells in eukaryotic organisms. So, hope you understood, and you see this is the structure which we have been following, and this is an animal cell is having a complex structure because this is a eukaryotic type of cell. Okay. So, we have been looked into a lot of times. So, I hope no need to go through all the organelles. So, we have gone through the organelles in detail. So, hope you understood the session, and this is a comparison of the structures of eukaryotic cell and prokaryotic cell. So, you see there are so many organelles in the eukaryotic cell, whereas in the prokaryotic cell you would see only few organelles. So, that was the main difference, we looked at it. Okay. Now, this is the topic of discussion what we had, and there are the differences, the very same differences what we have learnt here, following which you saw the presence of the prokaryotic cell with a cell wall, and there also you would find the presence of the cell wall exclusive to plant cells. Okay. And plasma membrane, we saw it was lying inside the cell wall, having the same character what we have learnt earlier, that is selectively permeable, stands same in the case of eukaryotic cells also. Whereas, the size you see 1 to 10 micrometers, whereas here the size range is 5 to 100 micrometers. And you see the cytoplasm, it is quite common in both the prokaryotic and eukaryotic cells. Suspension of organelles in the cytoplasm is also common, and you would see some of the different organelles like cytoplasmic inclusions and all are quite common for both the type of cells, eukaryotic and prokaryotic cells. So, this is to compare the different features. Okay. So, that was the discussion I ended up here. So, hope you understood the topics. You can get back to me with the doubts if you have any. So, thank you for having the live class here. Thank you so much.